Hello friends, welcome to my channel Creativity and Mindfulness with Subhamita. So today is 5th September 2021 and as we all know today is the Teachers Day for India and the day was nominated to be the Teachers Day in the great memory of Dr. Sarvapalli Radhakrishnan, who was a dynamic personality for his great contribution in academics and Indian politics. So today we will talk about some different aspect of teaching. In the last episode, in the last year, when we celebrated Teachers' Day, we did a talk show with numerous teachers, how they maintain their profession, how they help the students, and how actually they help in the psychological development, psychosocial development of students. Beside giving them academic support, how they provide the mental support to the students. So that was our primary target in the last year. This year, I have planned this program in a different way because we now all know that basic science research, rather research is a very important field, is an emerging field to be appreciated. This field is not a new field, but probably the appreciation of this field is becoming higher with the advent of COVID-19. So today what we are going to do, me and a few of my senior researcher friends, we are going to give you a hint or a small brief of how a true research is conducted. Why we are going to do so? Because nowadays we all see that there are several posts in social media, several blogs, several newspaper reports which are misleading. We do not know exactly which one to be trusted and which one not to be trusted. And this kind of dilemma is resulting into either in misbeliefs, anxiety, or in overconfidence. And none of this is good for us, especially in the present situation of the pandemic. So, with a note, rather with an objective to explain research in a very simple way to all of you, we are going to start today. And this is a very special episode for me because today I am joined by my seniors who have helped me all through during my PhD life. And two of them are from the same laboratory, same institute where from I have done my PhD as well. So in that way, it is also my way of paying sincere respect to my mentor, my PhD mentor, my postdoc mentors, all my teachers. And I believe that learning is a lifetime process. So every day I learn something from someone. So probably we do not give that acknowledgement to those persons in our day-to-day life, but learning is never separated. It's a process and it's a continuous process. So through this program I take the privilege to give my sincere respect to all the persons who in some way has helped me to learn something new. So have a happy Teachers Day and let's be with us in the discussion, rather in the talk show, that how research works and why should we talk about research. I have titled the program as We the Researchers Must Take. And today I am going to be joined by, as I mentioned, by three of my seniors. One is Dr. Anik Das-Bhaumik. He is now a senior analyst in the diagnostic services in CCMB, which is Center for Cellular and Molecular Biology. and his main work is related to diagnostics of different complex diseases, and he has a brilliant record of publications. He has published almost 46 research articles in international peer-reviewed journals. I'm very happy that he has given me time today for this talk show. And then I will talk about Dr. Orpita Chatterjee, who is now working in Nebraska Medical Center. She is working on effect of radiation therapy can be further enhanced in a positive way so that the patients get the benefit out of it. So basically her work is related to radiation, cancer biology, and other things. I think she will explain it much better in a much better way. She has also a brilliant track record of more than 18 publications in international peer-reviewed journals. And I'm honored to also have Dr. Pinaki Mondal. He is also a researcher and instructor in Department of Surgery, Nebraska Medical Center, University of Nebraska Medical Center in USA. So he is basically working on stem cell regeneration and how this stem cell regeneration and reprogramming can be targeted to treat to heal the damage from pancreatitis, which is also a division of pancreatic cancer. So definitely we are going to have some interesting talk from all of them. And Dr. Pinaki Mondal, he is also successful enough to have his food stamp in the world of research. He has also published many papers in international peer-reviewed journals. So why I mention international peer-reviewed journals? Because this is also a note of awareness that for when we publish something, so there are certain procedures, and when it is an internationally peer-reviewed journals, so that means it has been processed thoroughly with the specialist, with the expertised persons, and whatever the finding we read from the international journals, peer-reviewed journals, those are actually authentic and original research works. So that's why I mentioned everywhere international peer-reviewed journals, and probably this is a note that you can also take with you, that whenever you get some research finding, it should be published in somewhere, and it is better if it is published in peer-reviewed journals, and best if it is in internationally peer-reviewed journals. So with this small introduction to all these three researchers, I am delighted to say another name in this talk show, who is Wendrila Baisya, and she is actually the person who is mentoring the young students and motivating them for research. She is an assistant professor in Netaji Subhash Chandra Bose Institute of Pharmacy, and her work is related to pharmacological interventions, how the drugs are absorbed in the body, how they work, and what would be the best doses of a drug that has to be taken by a person. So it is more related to the application field of the research part. So basically we are going to have a very holistic approach to have all the persons from different fields, and I hope this discussion will be helpful to you in many ways. So please stay connected and feel free to ask whatever be your questions. Stay there. So with the seniority, I would first request Dr. Onik Dashbhomik to introduce himself as a researcher and what he is doing presently now, and then we will move on to the discussion.
Thanks for the introduction, Subhamita, and hello everyone. Basically, I started my journey as a researcher, but now I'm more into the diagnostics. But still, I believe research is everywhere. If you want to, want to add something, some innovation to your life, or something which can help to your society from your work, then it should be through research, because Without research, nothing can be done. Even any technology
Exactly.
development or everything, behind that, some research is there. No? So new ideas,
Yes.
innovations. So I started my journey. I after immediately after my post graduation in 2004 from Calcutta University, and I joined Manovikas Kendra in Kolkata. It's a beautiful institute of specially abled children, and I was fortunate to work with a very sincere mentor, my PhD guide, Dr. Kanchan Mukhopadhyay, and I learned a lot about genetics because I post-graduated in biotechnology, then I specialized into genetics. I worked with mentally retarded children and some other disorders also, interacted with them, and there are so many sweet memories. We used to counsel them. Uh, so, and my work was basically into finding the cause of this thing because these disorders is mostly genetic. So I think nowadays most of the people have idea what is gene and what is genetic disease, and it's a part and parcel of life. Because if you see the rate of genetic disease, especially in India, is very high, and there are different kind of genetic diseases are there, and it starts immediately after the birth of a baby. So there are several diagnostic methods to identify these genetic disorders, and there are many common genetic disorders are there in India. So my work started with identifying this cause to which the genetic cause which probably can be associated with the responsible for these diseases. So I work with mental retardation. It's nowadays called as intellectual disability and ADHD, and also Fragile X syndrome and the diagnostic part. So it's a very good learning. 7 years of my PhD life. I have seen so many. I interacted with the parents and the childs, and they have some deficiency, but they have some other special powers also. So this institute is a unique combination of both research and how to rehabilitate these children, help them to survive in the normal population of the society. So immediately after finishing my PhD, I joined in Hyderabad CDFD as a postdoc. Uh, this is also a CDFD Center for DNA Fingerprinting and Diagnostics. So I joined in the diagnostic division. I was fortunate to work with a very sincere mentor, Dr. Ashwin Dalal, and he motivated me to choose my path in the diagnostics. So there I tried to establish some of the genetic testing methods. Uh, in a cost-effective way, which can be done in countries like developing countries like India. And then I slowly moved to the sequencing. Means uh, sequencing means to identify the problem in the gene in molecular level. So in a single molecule, you are trying to identify what could be the uh, anomaly in our DNA. So there are several methods to identify, and there are these advanced methods like next generation sequencing. First, there were normal sequencing, and now next generation sequencing. So everything is next generation, right? So in IT field also, you will see first generation, second generation, third generation. So like that way, technology evolves. So the most advanced genetic testing methods is presently now is next generation sequencing. and microarray is another technique. So I worked on these two technologies. By the time I started, worldwide it was already there. India has just started. So I had to develop the pipelines, and this is mostly uh, the bioinformatics. So I have to develop when you are talking about the whole genome. So lots and lots of data is there. So to handle that data, you need bioinformatic pipeline tools to develop. And I'm not from bioinformatics background, but I had to
Yes,
learn.
and so Anita, that means you have learned this kind of data science techniques and all in addition to
Yeah,
your biological knowledge.
right,
So
right.
it's always a continuous learning journey that you have
Continuous
to
learning journey.
support
So the
yourself.
yeah, right.
Yeah.
So the technology evolves, and you have to update yourself with the technology. So now this is next generation, means it's the second generation. Now there is third generation, fourth generation. The technology sequencing technologies are coming. So you have to continuously update yourself in whatever field you are. So if you are stagnant in somewhere, then it's difficult to survive in today's uh, progressing science. So I worked on several NGS-based and microarray-based projects in my postdoc. I developed the pipeline and established the pipeline in the diagnostics. And almost 7 years I spent there, successfully published some papers also. Then after finishing my final project, I got a grant from SERB, means DST, Department of Science and Technology, Government of India, for independent research for 3 years as a young scientist. It was a project of developed by me. The idea was me and guided by Dr. Ashwin Dalal. So I worked on that project in my last 3 years of postdoc. Then after finishing that project, I moved to CCMB, Center for Cellular and Molecular Biology. So they have also a diagnostic uh, service. So that's in that I joined as a senior analyst, and in 2019, and now 2 years onwards, I'm working there. And professionally, I'm analyzing all the patients' data and giving them reports on these diseases.
Okay.
So you can see, yeah. Any questions there, Subhamita?
No, actually, I was just thinking that there is a very
Yeah.
common question from the people. Well, I am also asked sometimes, and I know the answer is quite complicated. But still,
Yeah.
I want to ask you because you have so much in-depth knowledge in this. So, when people talk about genetics, when we say this disorder is genetic, so most of the time it is the common notion that that generally people say, my father was taller, so I'm taller. My father is
Okay.
this, so I'm this. So in our case, not in our case, in most of the cases, the gene transmission doesn't go like this, right? It's a complex
Right.
trait and it has
It's a
different
complex
dose
step.
variations and these things. So I
Right.
just want to I just want you to address this point that while we talk about genetic diseases, it must not be of course it must not be a Mendelian inheritance, but I want you to address this one in a very general way, so that the misconception that okay, this is why it is so, my father was not like this, or why it is so, my mother has something different. So it's never is a crisscross, or it's never is a straight line arrow that you can draw uh, from your parents or grandparents. So here I want you to specify that how you should, like, how people should think about this part of the genetics, which is actually not very familiar to the people, to the normal people,
Yeah. So
especially with
basically,
reference to the diseases.
yeah. So you see, there are some diseases which are having direct Mendelian inheritance, right? So when we are inheriting some genes, one copy comes from father, one copy comes from mother, and there is a character developed in the child. Whatever is the dominant character, it will come up. And now there are some diseases which are multifactorial, means there are genetic diseases which where along with genetics, genes, there are other factors are there which some way control the phenotype, means the character. So it is in disease terms. So these are called as multifactorial diseases, or maybe polygenic diseases also. Means uh, if you see cancer, cancer is also a genetic disease. Some of the cancers. I'm sure Orpita and Pinaki will go into the details. But there are several pathways in the body, several biochemical pathways that may be involved. So the character comes, or the tumor develops, may not be always responsible for a single gene anomaly. It could be for different genes and different complex kind of anomalies that can cause cancer. And this is related to cancer. But there are some mental health diseases like autism, and you see this speech problem, hearing deficit. So there are multiple genes and some multifactorial. Suppose diabetes is not a mental health disease. Diabetes is that is also genetic, but it's a polygenic.
But diabetic amylopathy is there, which is also related to memory decline. And I think you also are going to say the same thing, like there are many genes as well which are common in between different diseases. So it is also about the balance of the dose of the genes. So it's not like that this one gene is for the one disease. So all the persons who are watching this program, please understand that genes are the basic of diseases. There are some genetic contributions in whatever way it may be, even if it is not a hereditary diseases. But when there is a sporadic disease also, so the gene has to be somehow, the protein that is having the problem, that is also coming from the gene. So in any way, the gene part is common for any of the diseases, but in general, what we say as a genetic disorder means it has a root of genetics, but that is not the straight one we usually think. So whenever you are being informed that your child or your family member has a genetic disease, that must not be so that your grandparents have to have this. And Anita also told one thing very, very important, which is multifactorial disorder, right, Anita? And there is a very big influence of the environment, which is very different for different persons. So in case of mental health, as Anita was telling about autism or ADHD, the environmental contribution is also very important. Anita, will you please say something about the epigenetic, which is actually very well explained by the environment, like epigenetic, in a very simple way. We just I know these are very complicated things, but people should know something about this thing.
Right. But I think Orpita or maybe Pinaki will be the ideal person to go through epigenetics. But I can tell you about the genetic causes, genetic variations in our genome that can be, there are some genomic variations. Those are lying into the simply the gene which is coding the protein in that part of the gene. But there are several regions in the genes which are not taking part in to code the protein. So they have other functions, regulatory functions, to make that protein correctly. So those variations mostly related to some methylation. These things is difficult to understand for the common people, but these kind of variations are called regulatory variations or non-coding variants. So epigenetics is mostly deals with those kind of variations, which can cause in this chromosomal anomalies and this cell division, all these things, it can cause anomaly. So I think the Orpita or Pinaki will.
Yeah, I'm going to Orpita D and Pinakida, but I just want to remind my viewers that you see with the one single charm genetics, we have at least talked about 15 minutes, and still we have not come to any conclusion, and we are not even to any pinpoint topic. So you can understand research is so vast. And when we talk about research, when we talk about the research knowledge to be translated in the social perspective, applicable perspective, so it takes a long time and the long phases. So what Anikta was talking about, genetics, the DNA sequences, which is very core to the molecular level, and there are different layers upon which you can go to the system. So it's more like cell to system. It's a big journey. It's rather a journey of a lifetime, I can say in a very simple term. So now I just come to Orpita D, my Orpita D, but she's actually Dr. Arpita Chatterjee. So Arpita, you just say about you have started your work in Down syndrome. You tried to integrate the breast cancer's pathways with the Downs and how they can be integrated with the SNPs and these things. As much I know about you, and therefrom you went to. Please correct me if I'm wrong. You went to NBRC, National Brain Research Center. You worked there for some times, and then you shifted abroad in USA. So how is your journey? And very specifically, I ask you that what you do now with this radiation, how after that you can cure the patients from radiation if you do the same. What I mean, I still will ask you to explain what you do, but still, so whatever you also do, you do at the molecular level. You also do not do it on the patients, right? You also do on some certain molecular pathways that you try to translate in the applicable format so that it can go to the clinicians and it can help the patients. So there is a very big gap between what we do in the laboratory and what the clinicians do with the patients. And this big gap is a very dark phase for the ordinary people to understand what happens where. So I will particularly request you to stress upon that part, that what we do is like what we do is translated to the clinicians, but how that process is, how long or how difficult.
So thank you, Subhamita, that you told me to talk about it, because it is a little bit hazy area for the patient and for the all the people, ordinary people who do not know about science. So the thing is, as you told, I started my PhD with same institute as Anikta. Anikta was my mentor there. And I started with Dr. Kanchan Mukhopadhyay.
Actually, you know, it's kind of a reunion. We three are from Monavikas Kendra. Pinakida is just few miles away in IICB. So it's like a nostalgic gathering for me as well. So yeah, please continue.
So the thing is, as I started with Down syndrome, and you mentioned about breast cancer. So the nice thing about Down syndrome is they are very much resistant to solid tumor. That was my point of interest there. That we all know that Down syndrome has a lot of intellectual disabilities and other hormonal problems, and other immunological problems, but they have a very unique thing that we don't have. That is, they have a very resistance to all the solid tumors, specifically breast cancer. So that was my point of interest, and I started from there why it is so, and I worked on that. And then I joined, as you told, that I joined to NBRC, National Brain Research Center, because in our Monovikas Kendra, we generally worked on genetics, like as Anikda already told, that we tried to identify the genetic SNPs, like genetic polymorphisms, what is the causing factor or maybe associated factor for the disease. And I did not have any hands-on training for molecular biology there, like other molecular biological techniques. So I wanted to learn that first, because it was difficult to move to some molecular biological lab if you don't have hands-on training. So I moved to NBRC, National Brain Research Center, to learn those techniques and how to design those experiments and how to think about those techniques. And then there I spent 3 years and I worked on glioblastoma. And then I moved here in UNMC. Here I joined this department of biochemistry and molecular biology. Is a huge department, and I joined with Dr. Rebecca Deegan, and she's a wonderful lady. Means she's a proper mentor, I can say. Means when I joined here, one thing I want to mention here, when you came from a different country, different culture, different way of working, the one thing I just identified, that way of working is totally different here. As you told that the gap between the clinics and the researcher, it is a lot more in India. Here that gap is not that big. So all the grants you apply, if there is any translational aspect, you can translate your research to the clinic immediately or very quickly, then you will be funded more. So you will think like
It's
that,
much open here.
much open, and
Yeah.
you can if you should think that your research, how your research should benefit your patient population directly. So that way you should learn to think. That what I learned, that not only the, yeah, definitely the basic science is encouraged, because basic, as Anikda told, that basic science is the father of all invention and everything. So you have to know the basic science, you have to do the basic science experiments, but there should be some applicability. If you So my research, when I come here, Dr. Deegan was working with prostate cancer and radiation therapy. So her research was how, when you put the radiation therapy on the patients, then they take the radiation therapy, but in long term there are a lot of different problems, like for the prostate cancer they will have some kind of urinal inconsistency and other pelvic region problem, fibrosis, and many other things. Like they do not take the uh, they do not take the therapy as well, means as desirable way. It is a lot of side effects are there. So if there is a lot of effects, you cannot put the desired dose on the patients. Like your dose will be less, and less number of dose you will give, so your cancer will not be cured that much. So that is two aspects of this research. One is put the radiation dose as desirable level, and another thing is just reduce the side effect of the therapy. So she was working on that, and our lab was working a nice thing with ROS, like the redox biology thing. And as you all know, for our lifestyle, changing lifestyle in nowadays, all the people have excess amount of ROS in our system, like reactive oxygen species, which can cause a lot of problems.
So you are talking about ROS, right? Because
Yes.
I say ROS because I find that most of the newspapers nowadays have a heading with ROS without explaining what is ROS. So ROS means the reactive oxygen
Oxygen
species, which is
species.
ROS.
Yes.
And so yeah, that is quite a highlighted term in case of anything, and even in the cosmetics walls also. Most of the cosmetics has this kind of uh, notations. Yeah, please,
So
Orchid, continue.
so thing is, ROS is kind of a, if you are chemist, you can think of a pH of a solution. So ROS is kind of like that. If you need that reactive oxygen species for your normal physiological reaction, but if you have more of it, then all the like bad, you don't want that. So ROS level should be in your proper level. And our lab, if you put radiation or any kind of chemotherapy, it will increase ROS, because you want to cancer cell to kill. It will increase the ROS, and by this high ROS level, your cancer cell will be killed. But as well as when you put the radiation on a tumor, the surrounding area will also be radiated a little bit. But you don't want the normal cells surrounding the tumor, you don't want them to die. You want them to proper save. So this off-target effect of all radiation and chemotherapy is mainly because of ROS, and you want the ROS system should be low in the normal cell, but in the cancer cell it should be high. That is our point of research now. And Dr. Deegan was working with a molecule which is right now clinical trial in different cancer, like at least 3 cancers. In even MC, we have started prostate cancer. We are going to start prostate cancer. We have already started with anal cancer also. This molecule can actually scavenge the ROS in normal cells, but not in the cancer cells. That's the beauty of this molecule. So it can increase the ROS in the cancer cells, so cancer cell will die with radiation, but it will not increase the ROS, rather protect the normal cell from ROS damage. So
okay, so Arpita, I just interrupt you for my viewers. So you want to mean that means you have some machinery, or some machinery means some chemical which can actually first distinguish a cancer cell and a normal cell. Normal cell means
yes,
a healthy cell and a cancer cell, and then it can have both. It can have the dual effect on cancer cell. It can increase the ROS so that the cancer cells can be dead in a very normal way. I
yes,
don't want to go into apoptosis and all those
yes,
things. So
yes,
it will be dead, and for the healthy cells, it will boost up its health.
yes.
So that means you are actually targeting in both the ways. In one way, you are trying to eradicate the cancer cells, and you are boosting up the healthy cells. So do you think that if you use this ones in non-cancerous cells, that means a person who is healthy and who is not having any carcinoma, can it be used in that persons to boost up their normal health, like to reduce the ROS? Or it is always has to be the balance between the contradictions?
So it should be in balance. You should not use this thing in the normal without any complications. So some research are there where they are using it as a, like, we have seen some anti-aging kind of effect on it, and also it is using as a cream for dermatitis. That is also very good. That is already in market. And also some this kind of thing. But I think if you don't have any problem, because this molecule has oxidant and oxidative property and reductive property, means if you have a cell which has a huge oxidative stress in it, it can damage that, and it as well as reduce the other part. But if you don't have any problem, I think it can enhance the health, but it is not clinically proven yet. So in public domain, I should not say that.
Okay, so that's a very good point, that anything that reduces ROS is not to be taken as a complementary medicine.
Because I want to mention one thing, because as the dietary antioxidant we use, like our curcumin and our other fruits and vegetables, for those kind of dietary antioxidant, you are taking as a very little amount, and you are consuming it, as absorbing by your body in a very little amount day by day. That is good for your health. But this molecule is the ROS scavenging capacity is very much higher than that, and your body will absorb more of it. So that amount is not good for your healthy cells if you are actually very healthy. So
Okay, that's a very good takeaway message for us. Yeah.
So dietary antioxidant you are taking every day, that's okay, that's good. Because
okay, so that means, so to summarize these things, this complicated things between what is good, what is bad, which one is what, I would say that people should actually be aware of what is they're taking.
I'm
Please.
Coming to that point, because as I after working with Dr. Deegan, I was interested in ROS, and then I noticed that huge amount of people in US and all over world, it is diabetic. Diabetes is a kind of an big problem, and diabetic people have lot of ROS in them, and that's why diabetic people are very much prone to different kind of cancer, different kind of other diseases. So if you are diabetic or obese even, you will have a lot of ROS in your body. So you should have to do something to reduce your ROS in the body, like do some exercise and take some good food like antioxidant rich food or healthy lifestyle. That will reduce your ROS, and it will reduce also your cancer probability. As Anikda was telling about epigenetic changes, and epigenetic changes are uh, very much on lifestyle, and that one of the biggest cause is ROS, change of ROS level in the body. And ROS change and the epigenetic change, and then the cancer or other disease, it is a very straightforward line. So you should think about your lifestyle always.
So that means it's the alternative medication is the changing the lifestyle, not
definitely
targeting the molecular pathway. And if we need, like, if someone needs to intervene with the molecular aspect, please consult the professional clinicians or the persons who know it, because as you can see, if a molecule has this capacity to distinguish a cancer cell and a healthy cell, and then can work on a different perspectives. And Arpita already mentioned that the diabetic patients has a tendency to develop cancers and other problems. And I can also say diabetic patients have a very high tendency to develop neurodegenerative problems with the brain plaques and everything. So you can understand, in the biological system, everything is so interrelated and intermingled. So if you want to target, okay, I'm getting fat and I'm obese, so I need to have a slimming pill, but you never know, this slimming pill can go to many part of your body depending on its pharmacokinetics. That means how it is absorbed by the body, how it is how it is taken away by the blood. So please, it is a message from Dr. Arpita Chatterjee, who is a scientist, and he's also asking you to change your lifestyle to be in the happier domain of your life, but do not intervene molecular things by your own. So thank you, Arpita, for telling these things. Actually, these are very important issues that are often, not highlighted. And people get misleaded, they think so many things, but they actually don't know what to do. And there are the social medias, the misleading advertisements and everything. So they say that it is tested, it is experimentally proven, but what is the logic behind this experiment, how it is, and it is always a dark phase. So thank you very much for telling these things. With this clue, I will come to Pinakida, who is now working on stem cells and how that stem cell regeneration can be effective for pancreatic cancer cells, that means which are already dead and damaged, and how can we regenerate the system with stem cells. So Pinakida, I would request you first to say your work in a brief, and you please focus on this part of the topic, that what are the experimental standards that every research should maintain, and that actually give the signal to be published in an international peer reviewed journals. And so whatever we see as technically tested, experimentally proven, and all those things, we are most used to with this kind of things nowadays, but those are actually not true researchers, and true researchers means it has a standard norm, scientific norm. So I would request you first to say what you do, what you have done earlier in Calcutta in Indian Institute of Chemical Biology, and then you please explain the standards, like how a process of research goes from scratch to publication, and how that is done, including the review process and everything. Yeah, Pinakida, up to you.
Yeah, I started working in IICB, Kolkata, and doing my PhD in 2006. And when I started doing my PhD, I didn't know much about genetics. We used to think that we know things about genetics when you're reading in MSc or in master's degree. But when I joined, I find out that I almost know nothing about genetics, human genetics. Started working, then Anita was working in Monovikash Kendra. Urpita was yet to join in Monovikash Kendra at that time. And while going to different seminars in Kolkata at that time, then I learned a bit and pieces about how the genetics things we read in the books and what is in reality. I can still remember very early days, probably I have joined 5 months or 4 months into my PhD. And my mentor, Dr. Sushanta Rajoguri, who is a really great teacher, and this is on the verge of Teachers' Day. I should mention that finding a teachers like him is like you'll find a gem that, oh, okay, so this is the way the science should work. So he's really
yeah. I was there as a summer training, so I also got this fortune to meet him, and he was so generous, and he was always very keen to make people understood what is what. So yeah.
What is what? Yeah. Before we do anything, he used to ask us that why we are doing this. The basic understanding of science is that why we are doing this, and what is the reason for it. As like everybody is doing experiment, I should start an experiment. Why I should start an experiment? Why we are doing this kind of thing. So I can remember one incident that I was presenting in Bengali, because that was a Bengali seminar. I was presenting in Bengali in Jalapur University about whatever the project he has given to me on oral cancer and cigarette smoking. So I presented whatever my lab was doing at that time, and one person stood up and asked me, why is that a lot of people smokes, but not everybody got oral cancer? And you are saying that cigarette smoking is causing the oral cancer. How come those people never got the cancer? I didn't have any answer for that because I was still a student. I was still searching for the answer. I didn't know what is going on. So I told him that we don't have a definitive answer for that. Still people ask me nowadays also that what should we do to avoid cancer? What should we do to avoid a certain disease? There is no definitive answer for that, and people got frustrated. Scientists does not know anything, kind of thing, you know. But the main reason I think today's topic also is the environment. The environment we are living in. What is the interaction of us with the environment? How we are interacting with the environment. So Dr. Rai Chaudhuri told me that Pinaki, go to different parts of our Bengal, try to find out who are the people who are smoking a lot smoking. They are smoking a lot, they are chewing a tobacco a lot. but they are not developing any oral diseases in their mouth cavity by inspection by a doctor. The doctor will inspect inside the mouth and they will not find any abnormalities, any leukoplakia or the benign form of the precursor form of the oral cancer. They will not find it. So go ahead and find out those people who are smoking a lot but are not developing any kind of abnormalities in their mouth. And you go to the cancer hospital and find out those people who are smoking a lot and developed a cancer. So you have two groups of people who have smoked a lot, used the tobacco a lot, developed cancer. Other people did not develop cancer. And you find out why these people developed it and why these people are resistant to it, or they haven't developed it yet. And our focus was to find out a genetic imprints of those individuals, what they have inside them. Now, as Anikdas told very earlier in this talk, that genome is huge. Genome is an entire genome. Looking the entire genome is a huge, gigantic task. So it's not easy. And when you are doing my PhD in 2006, it was even more difficult. So we looked as different parts of the genome which would be useful, we're guessing. Those parts we call DNA repair genes, the genes which are responsible for repairing any problems in the DNA. So Dr. Rajwari told me that you look at the DNA repair genes and find out whether those people who are smoking a lot but is not developing a cancer, has there anything in their DNA repair gene which is protecting them? Or the people who are developing the cancer has some kind of problems in the DNA repair genes which is giving them the cancer. Find out that. So my entire PhD was that we used to go and collect blood from those people. So it is starting from the scratch. This is the idea that why certain people develop cancer, why certain people does not. This is the thing a scientist wanted to do. I, as a student of the scientist, given that project, so my primary job was to go to different parts of the Bengal and to collect blood samples, 5 ml of blood samples in sodium EDTA vials. We transport back those blood samples to our research laboratories, isolate DNA from that, which is very simple process, can be done by a kit. So you get the DNA from different person. So let's say 20 people, 20 DNA from people who has smoked a lot in their entire 30 years of time, did not have any problems. And you have 30 samples, 30 DNA samples with you from the cancer patients who are smoking and got the cancer. Now, what we did that we tried to find out the DNA repair gene sequencing. We are not sequencing the entire length of the genome. We are sequencing only the parts where the DNA repair genes are there. How many repair genes are there? There are lots of repair genes. We focused mostly on 20 to 22 repair genes. So we sequenced only those 20 or 22 repair genes from the normal individuals, smoking a lot, did not get any problem, and people who are smoking and got the cancer. Then we compared those two and find out what are the difference between those DNA from this 20. It's not 20, I'm just saying 20 as a reference number. Then we compare and find out, okay, so there is an abnormality. There is something in that people, in the normal people, is not present in the cancer people, or something in the cancer people is not present in the normal people. And for that, we have to understand the scientific rigor or reproducibility, is that if you collect 20 samples from normal and 20 samples from cancer, is it enough to say that this is happening? No, it's not enough. So you have to calculate a statistically power calculation. I mean, you have to find out what is the power of my saying that this is the problematic problem in the DNA. How can I say that? So you have to go and say that out of 1.3 billion bases of DNA in human genome, I'm saying only one base causing the cancer. If I need to say that only one base out of 1.3 billion bases in human genome is causing the cancer, how many people I need to test it? How many people I need? And This is an observation. I know. I think I don't know what is the audience for this talk, but
Yeah. Actually, I just stop you here for a while. So uh, it's going in a very good way, because I wanted to strengthen this part of the thing, that when there is a discovery, or when there is something something we have found out, so that means it has to be in a population which can actually represent, rather it needs to be done in a particular sample size which can be represented in the population. So that means it is not an experimental result from 2 or 3 people or 20 people or 30 people. So it has to have a big confidence. When we are publishing something, it has to have a big confidence. Why I interrupted you and told you these things, because I think you also see sometimes that we and Orpita were talking about that nowadays many things have been there in the market with many kind of taglines, and even in social media also nowadays you will find many kind of research articles, or some cuts from the paper that are saying that this has been experimentally proved, or this has been experimentally done recently. Not very recently, few months back, I just saw that there was a sudden notion that regarding COVID vaccinations, that COVID-19, in case of COVID-19 in India, Covaxin is not adequately combating against COVID-19 vaccines, COVID-19 virus, whereas Covishield is doing. And then there was a controversy because it was done probably in some of the doctors, or some in some clinicians, and then there was a controversy, and the main person who was in charge of Covaxin, he argued that the research methodology probably was not uh, following the exact rule. Maybe there was some sample biasness that we generally talk about. So when it comes to the people do not know what is sample biasness. People do not know what is the sample size, in how many percent it has been replicated or repeated. But they ultimately come to know the bottom line. So here my objective to the audience is to make them understand that whatever you see in a general perspective is it may not be the same thing what is happening. Like, because I'm telling you that because when the term for second doses of Covishield that has been increased, there was an increasingly huge debate on why it has been done. So it should have been done from the first day, or it should have not been done. It is just it was taken just as a political scenario, but it was not. There was a serious research protocol for this. So as you were mentioning, this is quite pertinent to the talk, that why we need to do the sampling and sample size. So please, you can continue regardless of the thing that who are the audience, they will understand.
okay. Yeah, I mean, this is a very interesting way to understand it. Like, if I'm standing on a tall building and I'm throwing a stone when the people are walking on the street, what are the chances that if I'm throwing a stone targeting a particular person I wanted to hit, what are the chances that I will hit that person?
Exactly.
Because there's a lot of people are walking down the street. I'm standing a 10-story building. The chances, this is called the scientific, the statistical chances, that if you say that some incident happened, some result observed, is it happened just because of the mere chances, or is it really happening? So that thing we have to always calculate, that whenever we report something to the scientific community, we found this, we have to make sure that this is not just by some random result we got. This result we got repeatedly. This result we got most definitively within the constant of our sample population. This happened in Bengal. This happened in probably the south portion of the Bengal. This happened in oral cancer. This happened in the repair genes only. That's all right. But this really has happened or not? That we have to find out that really happened on. As for the COVID or cancer virus is not my expertise. I should not comment on that. But for any trials or any works or any results, people has to find out what are the number of samples we should test to say this is happening or this is working or not. For me, for that particular genome variants, I have to find out how many samples, how many number of individuals I should recruit to my study. I should explain to them that why we're doing this study. They would agree that they will participate in the study, and then we'll find out whether there might not be any difference. That could also be a possibility. There might not be any difference. But if there is any difference, is the difference is really happening or not? So instead of 20, we actually did it more than 2000. I am forgetting the exact number, but I think we have done it more than 2000 controls, people who does not have any cancer and 2000 people who has the cancer. And then whenever we got the data, okay, it looks like there is a portion of the genome which is changing. We need to go back to the community again, gather another 500 samples, fresh samples, not the same samples. Fresh new 500 people does not got the cancer. Fresh 500 people who got the cancer. Test it again to find out whether the result you got in the previous batch, you are getting the exact or the same result for this new batch or not. Then you report like this, and it has to be understood by the non-scientific community. That's why I'm pressing this so much. We report like this. We did an investigation on 2000 oral cancer people in southern part of the West Bengal with the age range of 35 to 65 who are smoking this amount got the cancer, and 2000 normal individuals from the southern part of the Bengal who are smoking for more than 30 years, age range of 35 to 65, does not have any abnormalities in the oral cavity by visual inspection at least. And we find out there is a difference. We tested this difference in another 500 cancer people and another 500 normal people and find out, yeah, the difference is still there. And we are reporting this difference might be a unique association with the oral cancer in the southern part of the Bengal among these individuals. So it's very specific. We are not saying this particular identification result is worldwide. Go ahead and
Exactly.
do it for, yeah, it's not like that.
Yes. So it is very specific and to the point. Why I actually mentioned about this COVID ones, because COVID is the most highlighted topic. People know many things about COVID with the blessings of the social media and these things. And actually, I know the research articles are also freely available, but I doubt how many actually can go and access this, because of course the research articles are not written in that term that everyone can understand. But still, I just mentioned those things because I can see there are many contradictory views among the people themselves, who actually do not know what has been done by one research group or the other research group, but with the conclusion, and which has been portrayed by the representatives, not by the actual conclusions of the articles, but the represented form of it has got such a good belief or bad faith that they start arguing, and this has led to some confusion, some worries, some misbeliefs and whatnot regarding vaccines and everything. So just to give that example, actually I refer to those two things, but this is actually for all kind of diseases, for all kind of inventions, that what is invented or what is told as a fact is dependent on certain conditions. So this is true provided this are there, and this is, and even the researcher who is telling this one, he also cannot say what will be the corollary of it, like this is true given this true, and may not be it is so straightforward sometimes that this is false because this is false. So it's a very complicated and very specific scientific things which are very difficult and challenging for ordinary people to understand. So what will be your message to the ordinary people, that how they should react to the new coming? Actually, this is a question open to all, how a normal the ordinary people should react to some information which is actually scientific information, but which is given in a totally I should not say non-scientific, but given in a totally different way. So how should people react to that? But before I come to this question, I first want to go to Wendrila, because when we talk about this kind of knowledges and everything, so she is the person who is dealing the students. So once a student starts thinking about in a new way or the analytic way, so she's the person who actually trains the students things. Yeah. So Indrila, as much I know about you have worked a little bit on some medicational aspects, and which is actually a little different from us. We all work on the molecular aspects and everything, and you worked on the clinical part of it, like how a medicine goes in the body and how it is. If I'm wrong, please make me correct. How it is absorbed, or how we should titrate the doses, which one is how much dose is effective for a person. You work on these things, right? You have worked on these things, and you also have publications in numerous papers. So for you, first I will ask that how simply you will say that the medication that is actually prescribed, so it has various effects on the body, even, and that is why there are side effects. No medication has it is not possible that a medication doesn't have a side effect. So every medications have some side effects. So how will you explain like this thing, that yes, it's normal that a medication, that a drug, that a prescribed drug will have some side effects? So how will you explain this one in a very general term, like for my viewers?
Yes, I am Indrila Boisho. Presently, I am by profession, I am assistant professor in Department of Pharmaceutics in Netaji Subhash Chandra Bose Institute of Pharmacy. So, as Subhamita told me, that my specialization is to design a dosage form of a drug. If the drug become a tablet or the capsule or injection, whatever it may be, to give it the better therapeutic activity with less side effect. My specialization is on that topic actually, to design or to decorate them the dosage form. Okay. Now, coming to the topic that as Subhamita told, non-scientific people. Actually, what we'll do after completion +2 level or higher secondary, the student come to us. আমাদের কাছে আসে, যেই দিন আসে, the first day of our pharmacy life, they don't know anything about the pharmacy.
কিচ্ছু জানে না।
The student, they don't know, কিচ্ছু জানে না.
Exactly.
Very few student, very least amount of student have very little
some knowledge of it.
When we ask them why you join the pharmacy course, maximum student go, my guardian, my brother, my sister
বলেছে
suggest me to do that subject. বলেছে যে আমাকে তুমি এই সাবজেক্টটা নাও, ভালো সাবজেক্ট। But what is pharmacy? They don't know anything. তো ফার্স্ট ডে আমাদের ক্লাস শুরু হয় with the WH word. Why, how and when. আমরা প্রত্যেক জায়গায় বলে থাকি why, how and when. কেন আমরা এই কাজটা করব, কিভাবে করব, এবং এটা করলে এটার কি হতে পারে, মানে প্রসপেক্টটা কি, এটার কনস কি আছে, প্রস কি আছে, কনস কি আছে, আমি কেন করব।
আমার করলে কি উপকারে আসতে পারে, করলে কি খারাপ হতে পারে, এবং কিভাবে সেটা আমি করব। সো ক্লাস উইল স্টার্ট উইথ দ্যাট যদিও আমি হার্ডকোর রিসার্চার নই, আমি এখানে যারা 3 জন আছেন আমি তাদের মতো রিসার্চার নই, বাট আমাদের রিসার্চ প্রোগ্রেসটা শুরু হয় হচ্ছে ওই সেই প্রথম দিনে WHO ওয়ার্ড দিয়ে। প্রোগ্রেশনটা শুরু হয় ওখান থেকে, যে আমরা কেন করব অ্যাকচুয়ালি। তো এইভাবে তাদেরকে আমার মেইন মোটো অ্যাজ এ টিচার, মাই ফার্স্ট অবজেক্টিভ ইজ হাউ টু লার্ন দ্য সাবজেক্ট। মিনস স্টুডেন্টকে আমাকে বলতে হবে, বোঝাতে হবে, প্লিজ তুমি ডিপে যাও সাবজেক্টটাকে তুমি ভালোবাসো, প্রথমে ভালোবাসো সাবজেক্টটা, যে আমি কেন পড়বো, মানে আমাকে এখানে ভিতরে আমাকে জানতে হবে আমি কেন পড়বো। ভালোবাসো, অটোমেটিক্যালি যখনই তুমি সাবজেক্টটাকে ভালোবাসতে শুরু করবে তুমি তার ডিপে যেতে শুরু করবে। যত ডিপে যাবে সাবজেক্টটার, তোমার তার যে এরাটা আছে, তার যে পাথওয়েটা আছে তোমার কাছে ওপেন হবে। দ্যাট ইজ দ্য স্টার্টিং অফ রিসার্চ, আই থিঙ্ক। এইবার যদি আমি বলি, তারপরে আমাদের কোর্স কারিকুলামে টোটাল, আমি জেনারেলি ইউজি অ্যান্ড পিজি ক্লাস নিয়ে থাকি স্টুডেন্টদের, ইউজি স্টুডেন্ট অ্যান্ড পিজি স্টুডেন্ট। ইউজিতে যেটা আছে কারিকুলামে, আমাদের ফার্মেসি কারিকুলামে, যে তুমি যে সাবজেক্টটা পড়ছো তার সিলেবাস অনুযায়ী আমাদের 2টো প্রজেক্ট তাদেরকে সাবমিট করতে হয়। খুব ছোট একটা থিওরিটিক্যাল প্রজেক্ট, মিনস অনলি রিভিউ অফ লিটারেচার, মানে তুমি কোনো একটা পার্টিকুলার ড্রাগ নিতে পারো, কোনো একটা ডোসেজ ফর্ম নিতে পারো, কোনো একটা ডিজিজ নিতে পারো, হোয়াট এভার মেবি ইউ লাইক, জাস্ট thoroughly review it and present it. One is that project, and one is practical project, very small practical project. You just take the animals for your study, or you use some instrument. Just তুমি নাও, তোমার একটা ছোট তুমি কোনো ডোজেস ফর্ম বানাও বা আদার ফার্মাসি ওয়ার্ক যা যা আছে তুমি সেটুকুনি করো, তোমাকে সেটা উইদিন 6 মান্থে তোমাকে সেটা প্রেজেন্ট করতে হবে। এটা হচ্ছে বিফর্ম কারিকুলামে থাকে। আর এমফার্ম মানে পিজি কারিকুলামে যেটা থাকে, থরলি 1 year project, as like you call me a mini PhD. That means you have to find out the review of literature, one particular topic, then all the you have to gather all the chemicals and the instrument, then you proceed thoroughly work, then result and conclusion thoroughly. As a teacher আমাদের তাদেরকে গাইড করতে হবে যে তুমি যে সাবজেক্টটা বা যে টপিকটা নিয়েছো সেটাকে তুমি বেটার এমফাসাইজে আরো বেশি করে তোমাকে জানতে হবে। এইবার প্রশ্নটা হচ্ছে, যেহেতু আমরা টেকনিক্যাল ব্যাকগ্রাউন্ড থেকে আমরা আসি, প্রথমেই ইন জেনারেল টেকনিক্যাল ব্যাকগ্রাউন্ড বলতেই আমি মানে খুব বেসিক সায়েন্স থেকে নই, আমরা টেকনিক্যাল ব্যাকগ্রাউন্ড থেকে আসি। তো প্রথমে টেকনিক্যাল ব্যাকগ্রাউন্ড বলতেই প্রথমে মনে পড়ে ইন্ডাস্ট্রি ওরিয়েন্টেড। ইয়েস, ইট ইজ ইন্ডাস্ট্রি ওরিয়েন্টেড সাবজেক্ট, কিন্তু ইন্ডাস্ট্রিতে যারা যারা যেতে চায় তোমাকে বেটার টেকনোলজি ডেভেলপমেন্ট করতে গেলেও তোমাকে সাবজেক্টটা ডিপে ঢুকতে হবে। টেকনোলজি নচেত তো সেই একই থরো প্রসেস চলতে থাকবে ইন্ডাস্ট্রিতে, ডেভেলপমেন্ট হবে না।
ইনোভেশনটা তো সব জায়গাতেই লাগবে, ইনোভেশন এন্ড রিসার্চের তো সমস্ত জায়গাতেই,
সব জায়গাতে। তো টেকনোলজি ডেভেলপমেন্টের জন্য, টেকনোলজি ট্রান্সফারের জন্য তোমার তুমি যেটাই জানবে তোমাকে বেটার অনেকটা মানে কি বলবো মানে অনেকটা ডিপার ওয়েতে তোমাকে জানতে হবে। তুমি আফটার কমপ্লিশন অফ বিফার্ম অর এমফার্ম ইউ গো টু FND, ফর্মুলেশন ডেভেলপমেন্টে যেতে পারছো, তুমি R&D তে যেতে পারছো বেটার টেকনোলজি ডেভেলপমেন্ট করার জন্য। অর আরেকটা যেটা আমি বলবো যে এখন এন্টারপ্রেনিয়র নিয়েও অনেক কথা চলছে, আমাদের গভর্নমেন্ট অনেক বেশি সেটাকে এমফাসাইজ করছে। তুমি তখনই একজন সাকসেসফুল এন্টারপ্রেনিয়র হতে পারবে যখন তুমি তোমার ডেজায়ার, তুমি যখন সাবজেক্টটাকে বেটার ওয়েতে জানতে পারবে এবং সেটা ডিপে ঢুকে সেটাকে রিসার্চ করবে, তখনই তুমি ইউ বিকাম আ গুড এন্টারপ্রেনিয়র অ্যাকচুয়ালি। তো এইভাবেই আমরা চেষ্টা করি আমাদের স্টুডেন্টদেরকে, মানে যে একদম মানে আমি নন সায়েন্টিফিক পার্সন থেকে, আমি যদি ওই টার্মটা ইউজ করি, যে নন সায়েন্টিফিক পার্সন মানে জায়গা থেকে আমরা তাদেরকে উইথ সাম সায়েন্টিফিক নলেজ আমরা তাদেরকে এইভাবে একটা পাথওয়ে দিয়ে মোটিভেট করার চেষ্টা করি যে হ্যাঁ তোমরা বেটার রিসার্চ, যত তোমরা বেটার রিসার্চ এরিয়াতে যাবে, যত তোমরা জিনিসটাকে বুঝবে, যত তোমরা সাবজেক্টটাকে বুঝবে, তত তোমরা মানে তুমি এবার তোমার ফার্দার চয়েস স্টুডেন্টদের যে সে কোন দিকে যেতে চায়, R&D তে যেতে চায়, FND তে যেতে চায়, না ড্রাগ কন্ট্রোল আমাদের যেটা আছে।
হ্যাঁ, মানে বেসিক যে একটা রিসার্চের মোটিভেশনটা সেটা শুরু হয় অ্যাকচুয়ালি। হ্যাঁ, এটা খুব একটা বড় কথা মানে বললে তুমি, বিকজ একটা স্টুডেন্ট তখনই তো রিসার্চটাকে নিজের প্রফেশন করবে যখন তার সেটাকে ভালো লাগবে। অ্যান্ড we all know like research is never a lucrative profession. If you talk about, I think my other my seniors would say the same thing. Research is never a profitable profession. It is only taken when you have a deep passion for it. Am I right, Pinakida, Orpitadi and all? Orpitadi, yeah, please continue.
So I have a question to Anikta and I have another question to Indriladi. That is, the question to Anikta is, means I don't know the scenario in India right now. So how much the basic science and the intermediate part, which is like diagnostic and the R&D in industrial level and the drug development level to Oindrila, these three things, how it is connected? It is, if there is any policy which is implemented newly from the DST or from the ICMR or something, Department of Health in India, please tell us about that. If there is any bridge between,
হ্যাঁ, মানে তুমি বলছো একদম বেসিক সায়েন্স থেকে কিভাবে অ্যাপ্লিকেশন হয় সেটা বেসিক সায়েন্স থেকে ডায়াগনস্টিক্স অবধি যাচ্ছে।
if there is any established pipeline, if there is any new policy implemented in India which we don't know right now, if you know, please enlighten us.
Yeah, please, Anikda and Indrila, please, yeah.
Policy means government is here. They are encouraging to this transfer of technology, means from basic science to the application field. There are several grants that are they have announced, like BIG and SPERSH, and so these grants are to develop to help a person to materialize their innovative thinking, what they have done in their basic science research or whatever in their study they have done, so that he can come up with something new and become an entrepreneur. So when you are becoming an entrepreneur, means you are applying your knowledge and doing something just like as a service or some product or something which are beneficial to the mankind. So these kind of grants are there because I'm working here in CCMB, a part which is called Untitled Incubation Center, CCMB, AIC CCMB. The mandate of AIC CCMB is to help the young entrepreneurs to help with their idea and fund them for a few years so that they can establish themselves and come up something new. Along with that,
And what is the means? I understood that there is some endorsement to the young generation to establish their career as an entrepreneur, but how they are finding their invention is going to the people of India, means how that is translated. Is there any platform for
how that is socialized?
Not socialized, for going to the patient directly.
How
so
the patients are getting benefit.
Clinical perspective.
Means how it comes to the general population. So actually this not only helping with the grants. When you are coming up with something new, so what they do generally, they directly bring it to the social media and through all other media so that it can reach to the general population and people can be aware of it. So if it is some clinical solution, it reaches to the doctors and other clinical medical persons who are involved, and from that it goes to the general person. If it is something product related, so they do some sort of this press conference and all, media release, so they make it so that it can go each, and they try to do it, but still I think this is at a very early phase. So a lot of more integrity is needed. So nowadays what happens is there are, it's a big country, right? And there are so much politics and other things are involved with this thing. So I don't know how, yeah. So how, কি বলবো, কতটা স্বচ্ছতা আছে এর পেছনে? I can see some backdoor things also. So
but there is still some kind of platform that you can get a patent of your finding, and then you can go to the common people to just to give your service to them, because
so this AIC thing is not only for CCMB. There are several institutes. Government has given this AIC, means there are IIIT AIC, and some other institute also get this auto incubation center. So this center also helps you. If you are coming something with the new, they will make you apply for patent, and they will look after all these things, how you are getting patent and how it is getting more and more to the
general
population. To the general
and because
population.
we saw in the in case of COVID thing, we got a co-vaccine, and other vaccination is also coming in India. So India can do it, but in case of, because government is endorsing for those vaccination earlier quickly, so that's why we got our own vaccine. But in case of other research, other disease research, we are not getting that much amount of people are getting patent, but after getting the patent, we are not getting that much amount of medicine in the market from India. So that is a backlog, I think. So that's why
backlog is there. Maybe Indila also can highlight more into the medicinal level.
This question is to both of you. So
commercialized level e seta asche, mane patient obdi seta পৌঁছাচ্ছে, that's a question. Now that's a question.
I just say one thing, like this from innovation to this patenting, it also includes a lot of legal actions, right? So whether the backlog is in, like, whether it is actually a backlog in that segment, or like it's a whole backlog in the process.
No, it's the backlog of the system. So it's very difficult. It is a very larger problem. So the way you can do research in Western countries, other countries, here the situation, the thing is a bit difficult and complex also. If you want to do something good and which will do some real good to the society, then you have to, a lot of chakkar here and there you have to do, because there are people there who can who will pull you down. So those things are there. But
but still we are doing quite.
You are doing quite, and there are honest people and young people, they are fighting, and in this way this country is progressing. So let's hopeful that something will just to give one example with what Indila was saying, that how these medicines are, why these are not coming, especially from this COVID-19. I am not a researcher. There are a lot of work is going on in CCMB, COVID-related researches and diagnostics. I am not involved much, but as a general common people, I don't know. I think you have seen there is iodine is used as an antiseptic agent everywhere, and there are some good research papers where they have shown that how this molecular iodine can kill within a second to few hours different doses from this COVID-19 in vitro studies, obviously. Personally, I am taking a medicine which is, I am not naming this medicine. It's very good, just like betadine. Betadine is, everyone knows. It's that povidone iodine solution. This this is a very Kolkata-based pharmaceutical company makes this medicine, very cheap medicine. Long term, me and my family are using. And my father is always he's a veterinarian. So he suggested this. You can take this in paper mode. I used to take it by putting into some drops in cotton and used to inhale. It's so good. মানে যাদের শুধু COVID-19 বলে নয়, যে কোনো সর্দি কাশি, মানে যাদের আয়োডিন সংক্রান্ত কোনো সমস্যা নেই, থাইরয়েড বা ইত্যাদি কিছু নেই, it does a remarkable job, but no one is there to promote this. People are dying, and there are some people from some other countries like Bangladesh, Pakistan. Bangladesh recently patented one molecular iodine solution for COVID-19, but people doesn't know. Even in serious cases, people are dying and no one is there to promote. Why? I think because these are cheap money. There is a political pressure from the higher medicinal, how the other costly medicines, how those medical companies will survive if this medicine comes. So these kind of things are there. So, but still, we are hopeful, and people are progressing.
So, Anika, as a representative from the scientific community, do you think that our country should take an action for repurposing of the drug, and also bridging bridging the gap between clinical and molecular science? This is
obviously there should be a. Here, what happens? One group do some research. They want to keep it secret. Other people they don't want to open it, and this is a common policy of each and every lab.
We are doing science, we are doing the research, we are getting patent, we are getting the good research paper, but as a whole to the country, we cannot serve them because we don't have the policy, right?
Right. There's no definite policy till now. Governments, they're promoting and they are encouraging people, right? But there should be some clear-cut policy that should come from the mandate that this is what you should do, this guideline.
Because we have the good research paper coming out, we have the patents, but people are not
after getting the patents. After getting the patents, this is just like a publication that I have a patent. But after that, what happens? No one knows.
That's I wanted to point out.
Yeah. Okay. So thank you, Wendrila, for giving that very good speech on how you motivate your students. So now, as I left that segment of research before with a question that, as researchers, how would you suggest? It's an open question, and you can all answer. Like, how would you address the general population who do not know about the research methodologies, who do not know how the research works, and what is told is wrong or right, and even to judge whether it is wrong or right, where to go, which website to find, and all these things to clear their doubts? What should they do? So it can be answered by any one of you, but it is for the people who actually are not from science background, but they are constantly having the inputs of scientific things in non-scientific way. But probably I should not use the term non-scientific, but the scientific informations in a non-scientifically modified way, I should say, because I can see sometimes that only one light is highlighted where the condition is not highlighted, which is a very important thing. So in this way, you please mention how a person should judge whether the information he or she is getting is actually usable for him or not.
For general people, and we have discussed, I and Arpita discussed it a lot over why people think about science. The basic understanding, the people who we are sitting over here, the guardians or the people who has educated us, they always think this is the last source of being viable. They first think that the first way of getting a good hold on your feet is become a doctor or an engineer. If you don't get that good score in the entrance examination, you go to the basic science. And as Wendrela was saying, that when the people come, they doesn't have any understanding because they doesn't know. They doesn't know why they are doing this thing. They wanted to get clear in the doctors or the engineer entrance examination. Now they don't know what to do. They doesn't have any guidelines to go anywhere. So whenever any scientists say anything or any kind of scientific reports appears in Anandabazar or any kind of regional or national paper, people has this kind of laid back attitude that the people in the science are not the jewel of the society. The jewel of the society are doctors, engineers.
And that's why, Pinakida, probably you have seen that even during this COVID, there are so many interviews for the clinicians, but for the researchers, I could hardly see anyone giving an interview, except the very famous and the very high-level scientists. But researchers are nowhere. Exactly.
Yeah.
Please continue.
Yeah, the reason is that when my son or my daughter, I do not seek their career as a researcher because I don't think that's a pristine profession. I think doctor is a pristine profession. So when I read something about a technology, about COVID-19 nowadays, or anything like that, cancer or diabetes or anything, I think the people who are working on these diseases are not the brightest minds. The brightest minds are already in other professions. So whenever I read about this news, I always have a skeptical mind inside me. Is this true? Is this right? Is this wrong? Those kind of things always in my inside, because I know that the brightest minds are not working in this field, which is wrong, which is completely wrong. Now, the brightest minds or the people who has a nice intelligence of pursuing their career is the persistent people, not the people who got an entrance examination. They are the intelligent person. Don't get me wrong. But the person who does not get the entrance examination, has a persistence for 20 more years to go to a direction, is also an intelligent person. So yeah, so the person who is pursuing a job is different, but pursuing a profession is different. Pursuing a passion is very different. Now, Anita can tell, Indira can tell, or Pita and Shubhamita, you can also tell. With the educational background we have, we are paid very poorly.
Absolutely.
Then the reason for staying here is what? The reason for staying here is that everybody can say that whatever they're working, they're very excited about it. Why this is happening? You know about it. So our main force of working here is the excitement. We normal people don't see it because they want to see the money.
Don't realize exactly.
Yeah, they want to see the money. Where is your nice house and nice cars versus where is your excitement about how a protein works? Who cares?
Or in another way, they are more interested in the cause effect. Like you have a disease, so why do you have the disease and what is the medication?
Right.
This is something that are concerns for the general people.
Yeah.
So that's why probably the clinicians are so hyped. Of course they do they do in the front line. We are not the front liners, but probably that is the reason why we are highlighted.
It doesn't matter who is in which line. It's not a queue. It doesn't matter. The thing is that
that's also true.
Yeah, the reason I was telling about scientific and people, whenever people read about something in the newspaper, this is their mindset. That's we established here that people does not believe in science in first hand. They said, oh, this might not be true. They will come, oh, I have heard this. They will come tomorrow in the news channel and say that the thing we told yesterday is not true. This is a good thing. If I say that this is the thing which will cure the disease and I stick to it, no matter what changes, this is bad. But if I cut
that is not science.
Right.
Science is always, yes.
Yeah, if I correct myself tomorrow and say, whatever I said yesterday was true, but today we get some new knowledge and we are correcting our course, that is a good thing.
And that what happened partially to the Covishield, AstraZeneca, that doses response.
Everything. Yeah. So
Yeah,
whenever people read something on the newspaper, their first course action about whether this is true or not true, believe or should not believe, is to contact the person who is doing the research.
Yes, exactly. A very good
They should not Google it. They should not search for a publication.
Yes.
They should not try to understand by themselves. It's not possible. They should contact a person who is doing the research and ask them what is going on. Please don't try to understand by yourself. We still don't understand the science properly. How can you will understand by Google it?
Yeah, and even if you try to understand that, that's a good way that you are trying to learn. But when you are trying to learn, you should not be judgmental. You should not learn something just to judge to the statement whether that is right or wrong. If you want to learn it, you learn it for your knowledge, not for the judgment. Yes, exactly.
This is a very good example. I'm going to say it in Bengali, if there is a Bengali person in there.
Yes, sure. We
আপনার ঘরে যদি কোনো সময় জলের কলটা খারাপ হয়ে যায়, এটা আমি অর্পিতার সঙ্গে কথা বলি, তখন আমি সবাই ইলেকট্রিকের ফ্যানটা কাজ না করে, আমি একটা ইলেকট্রিশিয়ানকে ডাকি, আমি একটা জলের মিস্ত্রিকে ডাকি। আমি বোঝবার চেষ্টা করি না গুগল করে যে ফ্যানটাকে কি করে আমি কয়েলটাকে সারাবো।
একদম।
কারণ যে লোকটা ইলেকট্রিশিয়ানের কাজ করছে, সে অনেকদিন থেকে করছে, সে হয়তো পড়াশোনা শেখেনি খুব একটা বেশি, কিন্তু সে অনেকদিন থেকে কাজ করছে, সে জানবে আর 5 মিনিটে সারিয়ে দিয়ে চলে যাবে। সুতরাং আমি যেমন ওই জাজমেন্টটা ওর উপর ছেড়ে দিই, ও যদি এসে বলে এই ফ্যানটা চেঞ্জ করতে হবে, হ্যাঁ করে দিন। ও যদি বলে রেগুলেটর চেঞ্জ করতে হবে, আমি কিন্তু জানি না কি হয়েছে, আমি ওর উপরে ভরসা করি। ঠিক তেমনি আমি যখন কোনো কোনো গবেষণা সংক্রান্ত খবর পড়বো, আমি সেরকম গবেষণার লোককে জিজ্ঞেস করবো, তার কাছ থেকে জিজ্ঞেস করবো, আমি কেন নিজে বোঝবার চেষ্টা করবো গবেষণাটা ঠিক না ভুল, এইটা বোকামি আমার মনে হয়।
এবং তার থেকেও এবং অর্পিতাদি তুমি একটু বলো, এবং মানে as Pinakida told that this is quite true that you should consult a person who is in this field. You please say that our field is like research field is a vast thing. So what you are working on, Ross, is probably 1% that I know, and what I am working on mental health is probably 10% you know. So even within the field of researchers, we have so much of knowledge that we do not know everything in that level. So how is it expected for a person who is not in this field and can understand something simply by Googling or by reading some blogs? So we are not discouraging anyone to do that, but what you say, like as Pinakida told, what is your viewpoint in it?
So that the example, like you are working on like some neurological disorder, psychological disorder, or psychiatric disorder, these three things are different. Neuron means
exactly
all three things are not the same. And I'm working on ROS and prostate cancer. That means I am not a very knowledgeable person about the pancreatic cancer. So thing is, if one normal person want to learn about something, there is a lot of way to learn it. One Pinaki said about just contact to the person. That is one to the point way to learn it. And if you don't know anybody who is working on that particular topic, I want to just say, you can go to PubMed. This is for your viewer. You can go to PubMed, and PubMed, at least you can get some abstract of the research paper, which many people probably will not understand because it is written in a scientific way. If you are not understanding the PubMed publication, then you can go to, if you know somebody who will understand that. Another is, there is some scientific blogs are there that is very good, because like Science Daily kind of thing, who gives you the actual source of what they're saying. So you can read the blog.
That are the big short blogs. Please mention that.
Exactly.
That are the big short blogs. Yeah.
So you can go to Science Daily kind of blog, which they explain the paper in a layman language, and they will provide you the actual link of the research paper. Then if you are if you want to go to that research paper, you can go through that link, but do not go to a personal blog, do not go to a normal newspaper writing and আমি বাংলায় বলছি খুবই ডিসঅ্যাপয়েন্টেড অন আনন্দবাজার, বিকজ আনন্দবাজারের যিনি সায়েন্টিফিক এডিটর আছেন, আনন্দবাজার এত পুরনো একটা পেপার, এত জনপ্রিয় একটা পত্রিকা, কিন্তু তার সায়েন্টিফিক এডিটর এত পুয়ারলি এডিট করেন জিনিসটাকে, তার সম্পূর্ণ মানে বদলে যায়, অনেক সময় ভুল ইনফরমেশন থাকে, হুইচ ইজ নট ডিজায়ারেবল ফ্রম আনন্দবাজার কাইন্ড অফ পেপার। সো প্লিজ ডু নট,
আমার মনে হয় এটা শুধু আনন্দবাজার না, আমার মনে হয় মোস্ট অফ দ্য ডেইলি সার্কুলার
like that.
Actually, they edit because they have to brief it from 500 words to 250 words.
Exactly.
And for doing so, they do not know where they are cutting just to make the sentence shorter.
I'm to disagree, Coach. I'm disagreeing with this part.
Yeah, please.
I think this is not the real reason. Real reason is the person who is editing the part exactly knows what the story is about, or what is the real story. It's not about Anandabhajar or any part. It's everywhere in the world. They exactly know what is the real story. But if you publish a real story which says COVID-19 is a natural normal virus, it's not going to create a surge in your publication. But if
it's
you
not
say
sensational.
Right. If
Yeah.
you say there might be a possibility it's a war created by some other governments, people will read it. So there is a difference. They know exactly what is the story, but there is a market of that story that also needs to be considered.
No, I am talking about like normal, not COVID-19 kind of, like
Anything for any
antioxidant
disease and
kind
anything.
of.
Yeah.
As I'm person from ROS lab, so I'm talking about antioxidant. So
Yeah.
take this antioxidant, you will be aging will be less or something, but that kind of thing should not, that's a tabloid thing, that is not a scientific
That's why I that time I told you that please
Yes,
say
Oindrila.
about ROS because
Oindrila is saying something.
Oindrila বলো।
আমি একটু ইন্টারাপ্ট করতে চাই, সেটা হচ্ছে তুমি অর্পিতা দি তুমি যেটা বললে যে অ্যান্টিঅক্সিডেন্ট, ক্যান্সার, নিউরোনাল ডিসঅর্ডার, এগুলো অনেক বড় টপিক, আমি এগুলোতে যাচ্ছি না। শুধু ছোটখাটো যদি আমি একটু জ্বর হয়, সাপোজ কারো জ্বর হয়েছে, যে ওষুধ বা জ্বর কেন হচ্ছে সেটা না বুঝেই পাড়ার দোকানে গিয়ে 10টা প্যারাসিটামল নিয়ে চলে আসে, খেয়ে নিল, পেটে খেয়ে নিল, উনি কিন্তু কাউকে জিজ্ঞেস করলেন না যে আমি প্যারাসিটামল, বা সে কি জানে যে হায়ার লেভেল অফ প্যারাসিটামল যদি আমি ইনটেক করি আমার লিভার ফাংশন ফেলিওর হবে, আমার সিরোসিস অফ লিভার হতে পারে, সে কি জানে? আমি তো তুমি যেগুলো বলছো অনেক হায়ার টপিক, আমার সেই জায়গাটাতেই এখানে যদি একটু গভর্নমেন্ট পলিসি গুলোকে যদি একটু চেঞ্জ করা হয়, যার যেটা ডেজিগনেশন, ডক্টর ডায়াগনোসিস করতে পারে, সে ডায়াগনোসিস করুক, রিসার্চাররা বডির ভিতরে কি হচ্ছে বা ড্রাগ পুরো জিনিসটাকে যদি রিসার্চ, যেটা বলছিল যে পুরো জিনিসটাকে যদি রিসার্চ ওরিয়েন্টেড, তারা যে তাদের কাজটা যে কোনটা করলে বেটার এফিকেসি দেবে, আমাদের মোটোটা কি, যাতে আমার অসুখটা সারে, বেটার এফিকেসি ড্রাগটা পাই উইথ লেস সাইড এফেক্ট, এটাই তো আমাদের মানে আমাদের মোটো। এটাই করি না কেন, মানে আমরা রিসার্চাররা যাই করি না কেন, আমাদের ডক্টর বলো, মানে আমাদের প্রত্যেক যে মেডিকেল ফিল্ডে আমাদের যারাই আছি, যে যেমন ভাবে আছি, মোটোটা হচ্ছে যাতে আমরা রোগী সুস্থ হয়, দিনের শেষে রোগী সুস্থ হয় উইথ লেস সাইড এফেক্ট। এইখানেই হচ্ছে ব্যাপার, যদি একটু পলিসি গুলোকে যদি ঠিক করে যে ডাক্তার ডায়াগনোসিস করতে পারে, সে সেটুকুনি কাজই করুক, রিসার্চাররা টোটাল তার যে রিসার্চের এরিয়াটা আছে সেই জায়গাটা নিয়ে রিসার্চ করুক। আমাদের মতো ফার্মেসি ব্যাকগ্রাউন্ডের লোকেরা যারা ওষুধ নিয়ে কাজকর্ম করে, মানে কতটা খাবো, কিভাবে খাবো, কখন খাবো, কতটা ডোজে আমি খেলে আমার কোনো অসুবিধা হবে না, এই কাজগুলো যদি সাধারণ মানুষের কাছে অব্দি পৌঁছে দেওয়া হয়, এবং যখন তখন ওই ওষুধের দোকানে গিয়ে দাদা দুটো গ্যাসের ওষুধ দিন, দাদা দুটো জ্বরের ওষুধ দিন, এইটা যদি বন্ধ করা যায়, এই খুব কমন ড্রাগ গ্যাসের ওষুধ দিন বা দুটো ওমিপ্রাজল দিন, দুটো প্যারাসিটামল দিন, এই জায়গাটাকে যদি একটুখানি কন্ট্রোল করা যায়, আমার মতে বেটার হয়, আমার মতে।
হ্যাঁ, এবার সেই কন্ট্রোলটাও কিন্তু একটু অন্যভাবে আনতে হবে। তার কারণ তুমি যদি বলো যে জ্বর হয়েছে, ঠিক কথা, প্যারাসিটামল আফটার আ সার্টেন ডোজ
it causes liver problems, liver function. Not only paracetamol, there are plenty of drugs that has these things, but there are few drugs that has to be available in the market in ease basis also. So the policy, yes, so the policy has to be made in that way so that it is not cumbersome for the people also to access to a drug. There are already the drugs that are not prescribed, that are not given without prescription, but there are also severe high dose antibiotics which are given without prescription. So the policy has to be in a holistic way so that it can ultimately give benefit to the people.
দেখো থিং ইজ আমেরিকাতে যেহেতু হেলথ কেয়ারের প্রচুর খরচটা বেশি, and people don't understand this necessarily. They think that America is a better healthcare nation. Is not, because it's very costly. India is much better as a healthcare nation because everybody can at least afford some medicine in India. So in America, paracetamol can be found in the general drugstore. যে কেউ প্যারাসিটামলটা ওখান থেকে পেতে পারে এবং ওমিপ্রাজল পেতে পারে। তার কারণ হচ্ছে যদি একটা গরিব মানুষের হঠাৎ করে একটু জ্বর হয়, সে ডাক্তারের কাছে গেলে তার বিল আসবে 40,000 টাকা, 50,000 টাকা ইন্ডিয়ান কারেন্সি, সে সেটা অ্যাফোর্ড করতে পারবে না, সেই জন্য তাকে দেওয়া হচ্ছে। বাট দ্য নর্মাল পিপল হ্যাজ এ সার্টেন ভ্যালু অফ আন্ডারস্ট্যান্ডিং, আমি এটা কি খাচ্ছি। সে ওখানে গিয়ে ফার্মাসিস্টকে জিজ্ঞেস করে আমি এটা কি খাচ্ছি, কদিন খাবো, সে সেটা জিজ্ঞেস করে। দে হ্যাভ এ জেনারেল টেন্ডেন্সি, তার যদি একটা বিশাল বড় সিটি স্ক্যান হয়, সে ডাক্তারকে জিজ্ঞেস করে এটা কি হচ্ছে, হোয়াট ইজ গোয়িং অন, and doctor has a responsibility to explain him or her what is going on. কিন্তু সুতরাং drug availability is not much of a issue probably. The more important thing which are all agreeing together is about an awareness. যে আমরা এটা কি করছি, মানে আমরা এটা কি করছি, এবং অ্যাওয়ারনেসটা তখনই তৈরি হবে যখন আমরা আমাদের ছেলে মেয়েদেরকে ভরসা করতে শিখবো যারা রিসার্চ করছে। আমরা ভরসা করছি শুধুমাত্র টাই পড়া, গাড়ি করে আসা একজন ডাক্তার, যাকে 30 জনের লম্বা লাইন, সেই ডাক্তারটা ভালোকে ভাবছি, বাট ইট নট নেসেসারিলি হি ইজ দ্য মোস্ট ফ্রন্ট লাইন, যেটা সুমিতা বলছিল, ইজ আ ফ্রন্ট লাইন, যে তাকে বলা হয়েছে এই ওষুধগুলো অ্যাভেলেবল, সে ডায়াগনোস করে ওষুধটা দেবে, দ্যাটস ইট, রাইট। কিন্তু যে ছেলে মেয়েটা জুলজি পড়ছে, যে ছেলে মেয়েটা ফিজিওলজি পড়ছে সাধারণ কলেজ থেকে, সে উইল বি মাচ মোর ইজি তাকে জিজ্ঞেস করা যে ভাই কোভিড-19 ভাইরাস বা ভাইরাস ব্যাপারটা কি, তাহলে কি জুলজির ছেলে ভাইরাস কি জানে না, একটা ফিজিওলজির ছেলে ভাইরাস কি জানে না, সে তো অনেক বেটার জানে, কারণ সেটা নিয়ে পড়াশোনা করছে, সে ফাইলোলজি থেকে শুরু করে সবকিছু জানে। সুতরাং সে তাকে অনেক ভালো করে বলতে পারবে যে একটা ভাইরাসকে নতুন করে জেনেটিক এডিট করে তৈরি করতে গেলে কি কি করতে হয়, এইটা হতে পারে কি হতে পারে না, এখন নলেজ অনুসারে কি মনে হচ্ছে, আজ থেকে 6 মাস পরে হয়তো অন্য কিছু মনে হয়, সেই জিনিসটা বুঝিয়ে
অন্য কিছু মনে হয়, এক্স্যাক্টলি।
হ্যাঁ, কিন্তু সেই জিনিসটা তাকে জিজ্ঞেস করা হয় না, জন্য আমার মনে হয়
হ্যাঁ, মানে এইটাতে আমার একটা মনে আছে, জাস্ট few years back probably, so I came to know about a patient who was suffering from dementia. So people asked me whether because people know dementia means Alzheimer's, but there are different kinds of dementia. Different dementia doesn't mean Alzheimer's, and forgetting memory doesn't mean Alzheimer's also. So there are different phases. So the person from his family asked me whether he is suffering from Alzheimer's. So I told I don't think this is Alzheimer's, because the way the symptoms have come, it doesn't look like Alzheimer's. Probably it is something else. So then the person got diagnosed. Then the person got tested for CT scans and MRIs, and there I also saw the scan report. So there was not the typical things, the features of Alzheimer's present, but of course with age there are some changes in the brain. So then I also told that no, this is not characteristics Alzheimer's, but yes, of course with age there is a dementia. So he is demented, but not Alzheimer's. So everyone from that family listened to me, but I understood that I couldn't convince them. And the next day they met the doctor, And I will not say the name and everything, but the next day the doctor, and the doctor just told them that he is going to have Alzheimer's. It's more like an Alzheimer's, and he's going to have. So I don't think the doctor also knows that this is not Alzheimer's. This is maybe a geriatric problem or whatever it is. It is not Alzheimer's. But when he is informing the patient, he is just informing Alzheimer's because this is very easy for him to illustrate. So this is also misinformation. When I told that, it was not taken into the confidence. But when doctor told that, and actually a contradictory thing, that was more convincing for them. So doctor also probably knows many things that they don't convey to the patient just because it will take some more time. It will take some more effort to make them understand. And for that reason, patients sometimes know the half part of it. Even I know there is a patient who was suffering from some liver problem, and that was a kind of cirrhosis which is genetic. Hepatitis some form, which is autoimmune kind of thing, right? So the doctor told that patient that you have মানে বাংলায় বলেছিলেন তো আপনার লিভারে বাত হয়েছে. So and that was fine, and he left with that answer. So after certain time, when that person was suffering so much, she was reducing weight, she was suffering a lot, and after that she couldn't sustain her life. So she never knew that what happened to her. When I saw the report, I saw it is like this, and then I also didn't know it is an autoimmune disorder, and then I checked and I saw, okay, this is an autoimmune disorder. So he is conveying autoimmune disorder as bath. So this is as arthritis, like arthritis. So this is something that we don't expect as well. So this is a level of miscommunication too for the patients.
Yeah, right now it's a good example which Anita right now told us, that it's iodine, which could be a good medicine for many of the respiratory disorders, right? If a journalist will listen to it, he will publish a report that it's a lot of billions of dollars people are spending, but iodine can be a cure of COVID-19, which is a misleading.
Yes, and that is a statement.
Right. Now, if as a general person, I read these stories, what is my first point of like evaluating whether I'm reading it right or wrong? I should consult. If
I want to talk one thing. I just wanted to, you just mentioned a term is story. So I want to convey this message that whatever you are reading in a newspaper, that's a storyline of that newspaper. It's not a scientific
but that
report.
is more convincing.
That is storyline versus a report. You need to understand the difference between storyline and a report.
Right.
So that's the point.
To understand the report, I know that Anida is my neighbor and he is working with the research. So I should contact him and he will explain to me that what is this
So
thing,
ultimately,
right? If I go to a doctor instead, the doctor doesn't have any idea about it. Doctor can tell you that which medicine is available for COVID-19. You can ask him or her, the doctor, that whether this COVID-19 is good for me. I have a high diabetes or those kind of things, and doctor will say, yeah, go ahead and take the vaccine, right? But if you really wanted to know about research, you have to talk with the researcher. Because
exactly.
what Anita was saying that Bangladesh already got the patent, right? So, and Anita I would request Anita to put some insight about reading stories in the news and validating, like
yeah, and that will be our takeaway message for today's program.
How do we validate? Orpita said about going to PubMed, which I am not agreeing with that, because it's happened to me when Donald Trump said that if you inhale some disinfectant, it will cure the COVID-19. It creates a problem in America. And I find out people are putting PubMed literature in the comment section saying that there is a report that disinfectant can kill viruses. And I was going to leave that comment, but then I thought that it's my responsibility to tell them they are wrong. So I said, if you read the entire report, you will find out this is not true. That's the thing. So Anita, please tell us about it, because we are talking and you have not made a
Yeah, and with that we will probably with that that message we will conclude that how to acknowledge or how to understand the research, or how to validate the finding or the report that we get often to be scientifically proven. Yeah, Anita, please.
In my opinion, the best way to validate it is to directly talk to the researchers. And somehow it may be difficult for the common people always to do that. But I have seen common people who has contacted either me or some of my colleagues here, asking so many questions about vaccines, and they have taken the number from the, you can easily get the numbers and the details if you search in Google, right, or in the social media. And I'll take the example of this iodine thing. Here in CCMB, one scientist is also checking the efficacies of the iodines in COVID-19, and they have seen remarkable effect of iodine of killing the COVID-19 in vitro studies. And I have given this medicine, what I was telling you, a sample, little bit of sample to them also, and they have seen the similar efficacy. And even if this has a better, because this medicine which we are using from my childhood, a unique combination of alcohol, iodine, and other Ayurveda medicines. So it's both Ayurveda and pharmaceutical chemicals are there. So now people ask questions, but here the same thing which Pinaki told. I usually recommended many of these people this thing, and this is a very less harmless drug, because it is also having Ayurvedic inputs. So some of the people in my neighbors, they are cautious, because since doctors has not recommended, so we can't go. I think it's just a simple, just like you take Vicks and this kind of menthols to inhale, no? So in that way, only you can use. So no, we use betadine, but this is not recommended. But if I tell, I told them that I have seen, my colleagues are doing research, but they don't believe. So this happens. But coming to the question of this Pinaki, I think the best way to validate these things, don't depend on this newspaper things. Just try to find out the researchers, will get the contacts and directly contact them. And we are happy here to give you some reply which may be useful to you. So this is
thank you. Yeah, thank you, Anita. Exactly with your line, we are happy to help you. We are happy to inform you what we know. Even if we do not know, we can ask our friends so that we can give you some better answers. But please, whenever you get some scientific story in some in daily circular, in social media, wherever, please try to validate that, and then only you believe on this. So with this small message from all of us, everyone has particularly told that please contact the concerned person. If it is on some other topics, please contact that expertise person, because he is working on that field. If it is really something with the treatment, the doctors are the best persons, you say. But when you are talking about science, when you are talking about research, please talk to the researchers. Please give them the due respect they should have from you, because without science, without promotion of research, nothing is going to happen. And with that message, I think we all agree to the same point, right? So with that message to all of you, today, let's start our journey of learning. And in that way, we all can teach each other So how to get the adequate knowledge on a topic? So we can always exchange our thoughts, we can always exchange our knowledge, and that's the way we can upskill ourselves and we can be together in the field. So in that way, with an open call to all of you to know whatever you want to know, please call us, please contact us, and believe in what is exactly happening in science. So thank you for being with us. Stay home. I should not say stay home because now lockdown is over, but I will again say that lockdown is over. Now we are coming back to new normal. Well, I know there is still a controversy between whether India is entering endemic or it is still in pandemic, and people are making stories on that still India is now in endemic, so there is loose in restrictions and all. But please, my dear friends, don't go out of the control, because there is some statement that India is entering endemic. Even it is endemic, it will not take a much time to again go back into that position if we are not in control. So please be safe, maintain the guidelines, maintain COVID protocols, and take care of your health. We will again come back with the same program that researchers we must pick. So thank you and bye-bye.